All right, welcome everyone. We will wait maybe another minute to allow folks to join. Okay, we see the numbers of attendees rapidly going up, so this should be a great session. So let's get started. So thanks everyone for joining us for this second NeLLi Symposium. On behalf of the entire organizing committee, I'm really excited to welcome everyone to this session entitled Updates on the Tree of Life, which is a topic near and dear to my heart, as hopefully it will be to many of you. So as a reminder, just in terms of logistics, for all of these seminars, there will be two main speakers, and we will follow those talks with a short Q&A. So we encourage you to please post your questions using the Q&A function in Zoom. And our moderator will then ask these questions to the speakers. In between the two main presentations, there will be 4 short talks that were selected from the submitted abstracts. We unfortunately will not have time to have a Q&A during this session, but at the end, we will have breakout rooms with each of the speakers for you to have a chance to meet with the speakers and directly ask the questions and discuss their research. So with that very brief introduction, I'm very pleased to introduce Greg Fournier. He is an associate professor of geobiology in the Department of Earth, Atmospheric, and Planetary Sciences at MIT. So with that, Greg, all yours.
Thank you, Emily. And also, thank you for everyone for joining us today and for the speakers who have contributed to this seminar. So I guess we can just jump right into things with our first speaker. That is Chris Rinke. He is an ARC Future Fellow group leader at the University of Queensland, Australia. And today he is going to be talking about some of his research that focuses on his field of study, which is genomics, taxonomy, and the evolution of archaea and environmental systems. So during Chris's talk, you can post any comments or any questions you have that arise during the talk in the chat, and I'll keep track of those and I can read them during the Q&A to follow. All right, thank you, Chris.
Thank you, Greg. I hope you can all see my screen. So it's an honor to present this team effort here today at the NeLLi Symposium, and let's get right into it. So what's the main motivation here? Today, I would like to convince you that it is important to have a taxonomy, a rank-normalized taxonomy that has quantitative criteria to circumscribe taxa from species to domain, right? And another question that goes along with that is one that I ask myself a lot over the years is, what actually is a phylum? What is a Archaeal, a bacterial phylum, right? Or any other lineage, a class or order for that matter. And I looked up to get the popular opinion, if you will, on Wikipedia. And there it says the exact definition of a bacterial phylum is debated, right? And that's true for the last decades, I would say. Goes on to say one popular definition is to use a 16S cutoff of 75%, right? And there are a few issues with that. First of all, it doesn't talk about archaea at all, right? Second of all, most of us have moved on the whole field. We're working with genomes those days. Many of them have no 16S. Plus, if you use like a fixed cutoff, right, let's say 75%, you don't really account for different evolutionary rates of different lineages, right? That brings us to the main issues we have with the current prokaryotic taxonomy. And I did use a tweet here. I tried to anonymize it, but you can probably still figure out who it is. Anyway, it's out there in the public domain. And it's the question was here, are there any good recent publications to show how messed up the NCBI taxonomy for microbes is? And this seems obvious to many, at least from private conversations. And it makes sequence-based taxonomy classifiers very challenging. So two messages in here. I guess most of us know that the current taxonomy is a big mess, and I don't want to blame NCBI necessarily for that, because NCBI is really a repository, right, where all our taxonomic ideas and suggestions end up. And yes, it is very messy. And if you want to automate the classification, then that can be a big problem. It's the same for archaea and bacteria. Slight differences though. I think bacteria main issue is because we had a phenotypic classification and we now had to transfer that to a phylogenetic one. So there are some issues with that, right? Archaea is slightly different because the classification has always been sequence-based. And if you go back in time, and most of you are aware of that, right? The archaea have been proposed in 1977 by Carl Woese and his postdoc George Fox. And as Archaeobacteria, they later updated the name to archaea in the 1990 paper, right? And that's where this tree is from. And yes, I know we are now closer to like a 2 domain of life than a 3 domain of life. But I think that that slide is actually very good to bring back to bring across the message that back then they defined like 2 lineages, right? Woese and colleagues, the Crane-Archaeota and the Uri-Archaeota. And this definition or this characterization of those lineages has shaped archaeal taxonomy for the decades after, I would argue, right? Mainly because Crane-Archaeota, that was a very tight group, a small group of extremophiles, right? Whereas on the other hand, you had Uri-Archaeota, that was a diverse group, phylogenetic, metabolically diverse, right? What happened then after that, more lineages were recovered in the Crane-Archaeota. Eventually, mesophilic Crane-Archaeota were found, and based on the fact that they are mesophiles, the genome sequence, it was then argued, oh, there should be a different phylum, right? The Thermoarchaeota. After that, it was followed by the Eiger-Archaeota, Bathy-Archaeota, Fastradi-Archaeota, and so forth, right? So these Crane-Archaeota were kind of sliced and diced over the years. Many new lineages were recovered for the Uri-Archaeota, right? But they were always considered to be a very diverse group. So all these lineages were dumped within the Uri-Archaeota, right? Another lineage, another lineage, another lineage. And that brings us to what we have today. That's a tree from a recent review paper by Baker. And it shows the NCBI taxonomy, but that illustrates the point very well, right? You can see here that this group here, if you can see my mouse, that is actually the former Crane-Archaeota, and they have been sliced into multiple groups here, right? And that's remaining. The Crane-Archaeota, and even in there, new lineages were proposed also as phyla, right? A few other lineages here, the Archaeota, deepened lineages, but the elephant in the room is the Uri-Archaeota, right? This phylogenetic behemoth, you can see it takes up in this tree about like half of the diversity in the archaea, right? And it's actually even worse than that. But my main message here is that we're not comparing apples to apples here if we talk about archaeal phyla, right? It's a very uneven application of ranks. Is that the only issue that we have? No, I want to point out 2 more in the archaeal taxonomy, right? One is if you ever make a phylogenetic tree, which I do a lot, and then you decorated it with the current taxonomy, the NCBI taxonomy, you will see something like this, right? It is actually very messy. So here that's a genome tree. I decorated with several groups in the NCBI taxonomy, and you can see they are kind of all over the place, right? For example, Nanoarchaeota here, they are down here, up here. So which is actually the real Nanoarchaeota? It's really hard. It's not very user-friendly, right? So my point is, there's a big inconsistency between the taxonomy we have and evolutionary relationships, right? Polyphyletic outcomes, and that is a problem, right? On top of that, you can see many lineages here, the ones in black, are only assigned in NCBI to archaea. So if I have a new genome, put it in my tree, it ends up somewhere here, I can say it's a new archaeum, but not much more, right? So it's not very helpful. So I would say the third point is that we have a widespread incomplete classification in our current taxonomy, right? To sum this all up, it's also true, that's why I call it prokaryotic taxonomy, major issues. It's also true for bacteria, right? We have uneven application of ranks. For example, the Bacillus, genus Bacillus, we had to split this up in, I believe, like more than 80 genera, right? In the bacteria, we also have inconsistency with evolutionary relationships, polyphily, widespread incomplete classification, the same for archaea and bacteria. On top of that, as I mentioned, there's no accepted standardized approach to assign taxa to higher ranks. That is, phylum, class, order, family, whatnot. And that leads to problems. So many, I just randomly picked one phylum described as Archaeota, right? And then like a little bit later, another paper came out and said, well, no, actually that is just a group in the order Thermoproteales. It's not the phylum, right? And our own research confirmed that as well. So did the authors then do something wrong here? Describing that as a phylum? Technically, no, because there is no accepted standard. So if someone finds a new lineages and for some reason decides it's a phylum, proposes that it gets published, it's part of the taxonomy, right? There we go. And that, of course, creates issues down the road. So now what do we do? Maybe we should just like sit back and relax. Maybe it's going to resolve itself, right? Is this going to happen? I don't think so. Many other people have the same opinion. One reason is we are very good in recovering new genomes, right? For example, metagenome sample genomes max, the numbers have skyrocketed recently. There was even a study by the JGI this year with, I think, like 80,000 new max, which is great. It's super exciting, right? But the problem is if we dump all this new diversity on top of this very wonky taxonomy we have, it's probably going to be collapsed and become unusable pretty soon, right? And I have a quote here from Ramon's paper, and he says that might end up in an unsolvable chaos at some point, right? So what is the solution? The proposed solution is the Genome Taxonomy database. What is that? It is an initiative to establish a consistent rank-normalized genome-based taxonomy for bacteria and archaea, right? And in the latest release from April, we have about 250,000 genomes. Now, I don't have time to go into detail, but in a nutshell, we take our genomes from NCBI, RefSec, and GenBank. We do a lot of internal quality checks. We figure out what is the type material. We get this information for LPSN, also NCBI, and then we create the GTDB database, right? We do dereplicate it based on a species level, which is roughly defined as with an ANI cutoff, 95%. And then we have a dereplicated database, and we then infer the tree, right? That means now we can start the curation process, automated and manually. The first thing we do is we normalize the ranks, right? To do this, it's based on trees, right? We use evolutionary distance or evolutionary divergence to actually achieve that. You can see here in this tree, right? When we calculate a tree, we have the inferred evolutionary distances. That is the branch lengths, right? But we're not using the branch length shown in purple here. We actually use a relative measure. And in a nutshell, the way it works, this relative evolutionary divergence, we call it RED. The way it works is we assign a 0 to the root, and all extant taxa get a RED value of 1, right? And all taxa in between get an interpolated value between 0 and 1. And what that allows us, actually, it allows us to account for varying rates of evolution, right? So for example, this lineage here has long branches, supposedly fast evolving, right? But the RED values only go from 0 to 1. If you have another lineage, like I say, this one here, short branches, slow evolving, again, the RED value goes from 0 to 1, right? So we account for different rates of evolution and therefore can compare across the tree. Now, if we use this and apply it to the NCBI taxonomy, you can see we get something like this. And you would actually expect that you have differentiation here, right? I should explain. That is the relative evolution divergence, the RED value here on the x-axis, and then you have the different ranks here on the y-axis. And you would assume that the RED value differentiates phyla from class and order, but based on the NCBI taxonomy, it's not really the case, right? It's a big mess. For example, you have the Archaeota. There are outliers of this group of phyla here. And on the other hand, you have very shallow phyla like Mar. That's the Archaeota, for example, right? So what we then do in GTDB, we have two options. We can find different internal nodes in the tree, or we actually assign those lineages to like a more appropriate rank. After many, many rounds of curation, we then end up with the GTDB taxonomy, and you can see actually it looks more like what you expect, right? If you use the relative evolution divergence, this measure that I just explained, the RED value, you get actually mostly a nice separation for the different ranks, right? RED values for the phylum, the class. We allowed some overlaps in some cases just to accommodate already effectively and validly published names. So we want to keep the changes to a minimum, right, in our taxonomy. Now you can see Archaeota, for example, they are now here, they're in order So I would say with this rank normalization approach, we can actually solve the first issue, the uneven application of ranks, right? Then the next one was the polyphyly, and the way we do this is automated, but there was also a lot of manual work. Is we first actually figure out what is the type, right? And that's why I'm always say or mention that it's very important to define the type if you define a new lineages. So we go for the type species, the type genome. An example here, Enigma-Archaeota subterraneum, right? We know that was the type is here. So we know that that is the group Enigma-Archaeota in that case. And this other green taxon here is a wrong, or most likely wrong assignment in NCBI, right? Similar for Nanoarchaeum, we have a type Nanoarchaeum accretans, which is in here, right? So we know that is actually the true Nanoarchaeota. The rank assignment assigned it to a phylum, so that is the phylum Nanoarchaeota, right? And then all those others that are assigned in blue here, the Nanoarchaeota, are most likely a wrong assignment in NCBI, right? So by doing this, we can actually clean up the taxonomy. It says here in red, if we have higher ranks that have no types, that is a big issue, right? For example, here, the Vos Archaea, proposed as Vos Archaea, no type was defined, right? So which are the Vos Archaea? Is it this group? Is it that group? Is it this group down here, right? That is a lot of almost like detective work to figure it out. We have to go back, see what are the first genomes compared to like 16S databases. So it is a very tedious work, so that's why I was emphasized, please propose a type if you have a new lineage, right? We eventually figured it out. That group here is the Vos Archaeales, right? And this group down here got a placeholder name. So that also solves the widespread incomplete classification. For example, we can say the same for the base Archaeales, right? We now know that all those genomes that are not defined in NCBI, they're just Archaea. We can now solve this and say they are base Archaeales, right? So that way, we can work on this incomplete classification in GTDB. Every taxon or every genome has a classification all the way from domain down to species, right? So, okay, overall, what does it mean? For the Archaea, this is a scary number. If you look at it first, more than 90% of Archaeal genomes require changes to the existing taxonomy, right? Sounds like a lot, but it's actually not that bad, because you can see the active changes when we actually went in there and we had to actively change a name. For most ranks here, class to species, that's just a little bit above 10%, right? Most changes are passive changes in blue here. That means we actually added information that was missing in the NCBI taxonomy. An exception more or less is here the phylum. We had almost like 70% change here, right? Why is that? Easiest to show this is VR3. That is the GTDB taxonomy, the R89 database, right? And I have here in orange, that is the NCBI taxonomy. You can see already that is species dereplicated, right? You can see that the Archaeota, NCBI phylum Archaeota, actually accounts for almost like 2/3 of the archaeal diversity, right? So we had to split this up, and based on the rank normalization in GTDB, we have the phylum Halobacteriota, Thermoplasmodota, Mesanobacteriota, and Hydrothermarchaeota, right? It was slightly different for up here, the Cranoarchaeota. You can see that is actually the Cranoarchaeota, the original group Cranoarchaeota split up into Bathyarchaeota, Thalmarchaeota, and whatnot. It was later, because it's a nice monophyletic group, right? It was later combined with the term tax superphylum, right? And this whole tax superphylum actually represents a phylum lineage in the GTDB based on the red value, for which we used the name Thermoproteota that has been proposed earlier. Similar to Asgard, the whole Asgard group represents like one phylum Asgardarchaeota, right? After we published the GTDB taxonomy, we of course interested in how is it received by the community. I don't have time to go into detail here, but overall, about the 30,000 users we had on the webpage, the good thing is that most of them are returning visitors, right? And then the GTDB-TK, that's a toolkit that allows you to download this toolkit, and you can classify your own genomes, and that had across platforms almost like 80,000 downloads, right? So I would say it was received very well. Of course, not everybody is on board, and a reviewer in our Archaeal paper summed this up very nicely, and I think those reviews are public now, so I think it's okay to share it with you. So the reviewer said here, right, I predict that this newly proposed systemless taxonomy will lead to resistance from 3 kinds of researchers, right? Number one, traditionalists who want to keep living the old days and who keep ignoring the wealth of microbial diversity that has been overwhelming us since the dawn of massive parallel gene sequencing. These researchers keep refusing using DNA as type material with the sole argument, we haven't done this in the past, right? And that's definitely an issue. And there were even like, I think there was a paper, a preprint like 2 years ago, that stated that all mags are chimeras because they were assembled from computer files, literally from computer files. That was in the abstract, right? So I think that's an issue, but maybe if you just wait 5-10 years, I think this issue will resolve itself. Another problem are people who named one of the taxonomy groups that are now being renamed. I don't like the term renamed because sometimes, for example, for the Ascards, we don't rename them, right? We just change the suffix, so Ascardarchaea become Ascardarchaeota. Anyway, he says here, I've seen this in conferences in which they also presented the study. People fight for the names they came up with, often without any scientific justification, but simply because they are emotionally attached to that. And I've seen this where people come up and say, yes, we really like your work, we really appreciate it, but hey, the group that I'm working on, that should really be a phylum because yada, 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 right? And I think that's maybe something that we all as community have to think about, because there is, especially in the Archaeal microbiologist community, I would say, there is this perception, this wrong perception. I would argue that a new lineage is only worth something if it's proposed as a phylum, right? And I think that we have to get away from that because that's simply not true, right? And I think that this message probably goes out to, I don't know, editors of journals, reviewers, all of us, right? We should really value a paper based on the overall scientific content and not if the new lineage is proposed as a phylum or a class or an order, right? So I hope that will change in the future. And then finally, there are scientists who have serious concern about the validity of the rank normalization we present here, right? And I think that is a very valid question or criticism. We have to address this, and we did this, right? In a nutshell, oops, I jumped ahead. One question we get a lot is, if you rank normalize, right, using the red value, doesn't it matter where the root is, right? Or an argument is, we can't even fix the messy Archaeal taxonomy because we don't know where to place the root. Is this an issue? I would argue no. Mainly because I haven't showed you this, but if we calculate the red values to normalize the ranks here, right? It's not a fixed root. We use a system where we root on the major phyla, right? Which are defined as having 2 daughters. In that case, it's having 2 classes, right? We root on each of them. We calculate the red value, and then the final red value is actually the median of those multiple rootings, right? And that increases the stability of the GTDB taxonomy. If we ever find out, if we ever agree as a community, I don't know if we ever will, on the roots in the Archaea, right? How does that change? Well, it's still fine. So we tried this, right? If we go for a rooting scenario, and I think that might be the most widely accepted now, where we root it at the deep end group in the Archaea, right? You can see if we do this, if we force GTDB to accept the fixed root, there are slight changes, but overall, the different intervals for phylum, class, order, and family are still preserved, right? Also, if we tried another route that was proposed recently by a French group, we root within the Archaeota, right? The same thing happens if we force the fixed root. The main pattern is still recovered. So I would argue GTDB is independent of the root. If we ever agree on a fixed root, it should not lead to main changes. Another question that we get is, okay, but doesn't it matter what kind of, your method is tree-based, right? Doesn't it matter what kind of markers you use, what inference method you use, what alignments you use? And yes, it matters for the overall tree topology, right? But we are not interested in the overall tree topology. We're only interested in the taxonomy, meaning only in the internal node on which we put taxonomic labels on. And for this, we only use highly stable nodes, meaning a bootstrap value of 90% or above, right? And those nodes are very stable regardless of the method. We compared this here in this graph. So you can see here, those are the different markers that we use. For example, 122 marker genes, then RB1, RB2. Those are ribosomal proteins, 16 ribosomal, 23 ribosomal proteins, the 16S, 53 markers. And that's the result for the phylum. You can see in green, that is monophyletic as shown here. That's what we want. In orange, it could be that the phylum become polyphyletic, right? You define a phylum here, then another group jumps in. That's of course not a desired outcome. We also introduced operational monophyletic, and that is mainly for very large groups, right? So for example, here you have like this group has like 24 taxa, and then another taxon jumps in here. Or the other option is you maybe lose one taxa, right? So that doesn't make the whole group necessarily polyphyletic. We call this operational monophyletic because it's still very close to being a monophyletic group. And overall, you can see on the phylum level that except for the 16S, right, we had more divergence here. That was mainly because a lot of genomes max have no 16S, so the number of taxa was way lower here, right? But overall, more than 90% of those phyla that we proposed are monophyletic or operational monophyletic. We then extended this, right, to a whole different range of markers, different methods, IQ3, XML, phylobase, right? Different alignments to reduce compositional bias, fast evolving sites. And overall, you can see that this phylum, class, and order, that overall more than 90% of those taxa are actually stable, right? Regardless what kind of approach you use. And I think that that's actually a good outcome, at least a reassuring one. One more thing I wanted to show is people ask, okay, how does the GTDB scale, right? How stable is the taxonomy? And we also have to say there never is going to be a final taxonomy that's not going to change at all. That's going to happen, but GTDB is overall very stable. We simulated this, actually, Pierre did it in our team. He said, okay, why don't we go back and see how many of the genomes we have right now were present in 2015, 2016, 2017, right? And then we actually ran the rank normalization approach on those small data and see how that changes, right? To simulate this expansion that we had over the years. And if you do this, you can see the results here. Those are the different data sets, 2015 to 2018. And overall, right, again, more than 90% of the taxa, phylum, class, and order that were proposed in GTDB are monophyletic and operational monophyletic. So I think that's very reassuring. If we keep extending the number of taxa, or the number of genomes, sorry, I should say, and number of taxa in the database, right? It should still scale very well. Another slide I wanted to show you, but I'm probably going to skip this because I'm running out of time. We used the GTDB to reanalyze analyze the Archaeal, right? Because a lot of lineages were proposed recently, but I'll maybe just jump ahead here. Then I just want to point out before I finish that if you propose, right, a new lineage, please include type material. And I would like to refer you to this paper that was mostly spearheaded by Marcia in our group. And there we actually provide step-by-step and examples how to define actually type material for your new group, right? To sum this up, probably I would say that's my call out to the community, right? If you name something, in this example, I named an Archaeal phylum once, right? What did you call it? Bob, right? Please do not do that. Please do not call it Bob and walk away, right? That makes our work very, very difficult. If you want to stick with Bob, right, I encourage you designate a type material, right? So in that case, you would call it Bob Archaeum Bobby, right? That's the species. Then you have the genus Bob Archaeum, and from there you can propagate the name up the ranks, Bob Archaeaceae, Bob Archaealis, Bob Archaea, right? And if it actually is a phylum, and hopefully you use the GTDB to analyze that, then it would be Bob Archaeota, right? What that does is we have a type. So if for whatever reason this phylum should become polyphyletic in the future, right, we always know where the Bobs really are because we have a type to go back, Bob Archaeum Bobbii, right? So I guess that's my biggest take-home message. And that applies if you use the GTDB taxonomy. Yes, that would be great. Even if you use NCBI taxonomy, please describe type material, right? Then I think I'm gonna finish here. I just want to point you to the GTDB website. That's where we have the taxonomy. You can search it there. You can compare between GTDB and NCBI. We now also have a form. Most of the user requests and questions, we still answer them per email, but we try actually to direct people to the forum so the answers are out there for everybody to use. Then finally, GTDB-TK, it is a software toolkit. I mentioned that if you want to assign a taxonomy to your own genomes, you can download it. You can get it here, and there's also online support if you have questions. Okay, with that, I'm at the end of my time slot. I just want to thank the GTDB team and all international collaborators, our funding resources. I want to point you to Donovan's talk. He's going to talk about the ongoing census of Bacterial and Archaeal in the GTDB, the latest updates. And finally, want to thank you for your attention.
Thanks, Chris. We are out of time. We have maybe 1 minute. So I'm going to very briefly ask 2 questions that were posted in the chat. The first is from Robert Evans, and he says, it seems that some new classifications, the groupings are unranked. What are the reasons to call clades by special names like kingdom or phylum or family?
I'm not quite sure if I followed the whole question, but in GTDB, right, we actually assign a taxonomy for all the ranks. So we basically go from domain all the way down to species, right? So if you look at the GTDB taxonomy, if you look at your genome, it will have a whole classification for all the 7 ranks, right? So we try to be consistent here. In NCBI, it's slightly different because it's a repository, right? If people submit something and you submit your genome as Archaeal, whatever, it might be assigned to Archaeota, but all the intermediate ranks from like in that say class or the family are not filled out, right? So there are big holes there, and GTDB tries to actually fill those. So we have a whole 7 rank taxonomy for all entries in the database.
And our other question is from Reka Seshadri, and they ask, what is the recommendation for designing a type?
That is a good question, and that's why I pointed you to the paper. The details are in there, but overall, I would say it's the quality is the first criterion, right? There is actually the MEMAG and MESAG paper that was published by Bowers from JGI, and they actually specify a quality criteria. Like it should be more than 90% complete. You should have a 16S in there, tRNAs. So I would say first we need a good quality because that's right. We also want to future-proof this as much as possible. So good quality is the first one, and then after that, I will point you to the paper, and you find like more details in there.
Great, thank you. And we'll just jump right to our first short contributed talk. So before then, just a reminder on length. So the shorter talks are. Emily, what was that again there? 7 minutes?
Yes, they should be 7 minutes. And unfortunately, we don't have time for questions, but we encourage everyone to join the breakouts afterwards.
Thanks. Right. So, and first we have Donovan Parks from the University of Queensland. Donovan is a consultant working with the Australian Centre for Ecogenomics on the Genome Taxonomy database. He'll be discussing the growth of bacterial and archaeal biodiversity within this taxonomic framework. So Donovan, whenever you're ready.
Yeah, thanks, Greg. Can you just confirm you can see my slides? I'll take that as a yes. All right, yeah, so this will be just basically a quick overview on the current state of the Genome Taxonomy database, or GTDB. So in case there are some people joining us late and missed Chris's talk, the GTDB is an initiative to establish a phylogenetically consistent, rank-normalized, and complete genome-based taxonomy, specifically for bacteria and archaea. So it's phylogenetically consistent in the sense that GTDB taxa are defined to be monophyletic on a concatenated protein tree. It's rank normalized because species are delineated using average nucleotide identity criteria, and higher taxa are normalized. So ancestors of equal rank will have roughly coexisted at the same time in the past, and that's done using a measure called relative evolutionary divergence. And finally, it's complete because all genomes within the GTDB have assignments from species up to domain. So I'll begin just by taking a look at the growth of the GTDB since its inception in November 2017. So what you're seeing here is just a line plot showing the increase in the number of taxa broken out into different taxonomic ranks. So from phylum in light blue here up to species in red. So you can see that there's been a very substantial growth in the number of species in the GTDB. Now, I will point out here that in release 89, so at the 20-month mark, that's where we introduced species clusters based on average nucleotide identity. So before that, in release 86, there were many genomes that simply had no species classification. So a lot of growth in the number of species in the GTDB between 86 and 89 is because we've clustered everything into actual species. However, you can see that there is still a rapid increase in the number of species even past release 89. Now, as you might expect, the growth in higher ranked taxa is of course slower, but you can see that it's still increasing from release to release. Now, another interesting way to look at the GTB data is to investigate what the actual source of taxonomic diversity is. So this is specific to the current release of the GTB, which came out in April this year. And what these bar plots are showing you is taxa where the genomes comprising that taxa are isolates, mags, and potentially sags. So that's what's shown in blue. The orange bars here indicate taxa where all the genomes we currently have are actually isolate genomes. And the bars in green here are indicating taxa where all the existing genomes in that taxa are actually mags and potentially a few sags. And what's interesting to see here is that for both bacteria and archaea, the majority of taxonomic diversity is indeed in taxa where currently our only representatives for those taxa are mags and potentially a few sags. Okay, so now I'm going to focus specifically on species within the GTDB and how many of them are what we would call discrete versus fuzzy species. So I'm just going to illustrate what I mean by these two types of species with a pair of examples. So what I'm showing you here is a pair of Enterocloster species. These are closest neighboring species within this genus. And this is a clear example of what I would consider to be a discrete species. That is, the genomes within these two species are well separated from one another in terms of their average nucleotide identity. And what that really means is that there's no ambiguity in terms of what species a given genome belongs to. Now we can plot that out in terms of the interspecific and interspecific ANI between all these genomes. That'll give you the histogram I'm showing you here on the right, where you can see that we have this nice genetic discontinuity, again, showing that genomes in these two species are well separated from one another. Now we can contrast that with what I think is a clear example of a fuzzy species. So this is two Bradyrhizobium species. Again, these are closest neighboring species in this genus. And here you can see that these genomes form much more of a genetic continuum. In fact, it's a continuum to the point where the two genomes here that I've connected with that black arrow, they have a very high ANI and are actually more similar to one another than those genomes are to other genomes within their own species cluster. Now, again, you can plot out the interspecific ANI values. As you'd expect in this case, you get a lot of intermingling of those values, again, showing that we have a genetic continuum. And what's really important here is that exactly what species you're going to assign a genome to really depends on the exact set of rules and criteria you're going to use to delineate species in sort of this region of genetic diversity. Okay, so that's sort of conceptually how we can think, or how I think about discrete and fuzzy species. Now it's nice to put some actual quantitative definitions on this so we can sort of enumerate how many of these species exist in the GTDB. So for that purpose, we're defining a discrete species to be a species that contains no genomes with an interspecific ANI value greater than 94% to a genome in another species. So that is, all the genomes within that species are sort of quote unquote far away from the genomes in other species. In contrast, a fuzzy species is a species where one or more genomes has an interspecific ANI value greater than or equal to 95%. So that is, there is a genome in that species that is sort of very similar to a genome in another species. So we can apply that those definitions to the latest release of the GTDB. So that's release 202. And there are 35,000 species clusters that exist within genera that have two or more species, so where we can reasonably define closest neighboring species. And for this analysis, I'm specifically looking at GTB species that are defined with an ANI circumscription radius of 95%. So this is by far the vast majority of GTB species and what we would consider a typical species. but there are some exceptions in the GTB, such as E. coli and Shigella. So I'm just ignoring those because it's sort of a separate discussion. So we just look at these typical species. You can see that 90% of them meet the criteria for being discrete, 2% meet the criteria for being fuzzy, and then there's 8% that fall sort of between those two and would depend exactly how we want to define discrete and fuzzy. Now, what the take-home message here is that indeed there already are fuzzy species within the GTB. However, the vast majority are clearly discrete. What's an interesting open question is whether, as we discover more and more strain diversity, are these discrete species going to tend towards becoming fuzzy species? And then we'll have to have an interesting conversation about what it means to break genomes up into discrete species clusters. And indeed, there is really a continuous landscape of genetic diversity. All right, so with that, I'll thank you for your time. This is the wonderful GTB team that I have the opportunity to work with, and I will indeed be at the breakout session later to answer any questions you might have. Thank you.
Great, thanks, Donovan. And yeah, everyone, be sure to check out the breakout room sessions to ask any questions from any of the shorter talks. So now we go to Anthony Kohtz from Montana State University. So Anthony is a graduate student at Montana State in the lab of Roland Hatzenfilcher. He studies archaeal diversity and methanogenesis in Yellowstone National Park hot springs through metaomic and cultivation approaches. And are you all set to go?
Yeah, can you hear me and see my slides?
Yes, perfect.
Cool. Yeah, thank you. Big thank you to the organizers for putting on this awesome symposium and giving me a chance to talk about some of my work. So a lot of the work that we do in our lab takes place in Yellowstone National Park, which is a great natural laboratory because it contains about 14,000 geothermal features, which equates to about 50% of the global total. And so we're about a 2-hour drive away from a huge number of geothermal features. And these are great to work with for studying diversity because they're often tractable due to their often extreme conditions relative to other systems, for example, like soils. And there's a wealth of diversity housed within the park. And this is evidenced by the fact that many archaeal lineages were first described in Yellowstone National Park. For example, the Geo, Hades, Core, Mars, and Odin Archaeota. And so in the Hatzenfilcher lab, we're interested in exploring new archaeal lineages within hot springs. And we do this through culture independent as well as culture dependent methods. And so in a metagenomic survey of a couple hot springs across the park, including Washburn hot springs, as well as 2 unnamed features in the lower Culexarchaeota basin, we found that there were several 16S RNA gene sequences that formed a unique cluster within the Taxonomy. That's indicated on the right here in red, where we're indicating the Culexarchaeota. And we decided to name this after the Culexarchaeota basin in Yellowstone National Park, where we first detected these organisms. And so when we expanded this analysis to other public metagenomes, we were able to recover 10 mags from 6 different samples. And this included a variety of terrestrial hot springs, as well as from marine hydrothermal sediments in Guaymas Basin. And as you can see on here, all the dots are metagenome samples, and the triangles are where we detected a 16S RNA gene relating to this lineage. And we found that we only detected them in high temperature environments. So while they are widely distributed, it does seem that there's a very narrow range of temperature and pH that they thrive in. And these are also low abundance populations, which is likely why they were overlooked and not many representatives were available. And so when we take those 10 mags and do a phylogenomic analysis, it indicates that the Culexarchaeota are a sister lineage to the proposed methanogenic Bristol. So that's indicated on the left here with the Culex in red, and we can see that there's a robust support for the relationship between Culexarchaeota and Bristol. And as Chris and Donovan just outlined, with the GTDB classification, the phyla of the Archaeal are combined under Thermoproteota, and they're downgraded to a class. So in this example, the Archaeota were reclassified to Methanomethylacea. And so the Culexarchaeota could be folded into the scheme by reclassifying them as a class, or the Culexarchaeota. And so that's a phylogenetic approach for distinguishing the Culexarchaeota from others, but we also wanted to look at other ways that we could distinguish Culexarchaeota from other related archaeal lineages. And so we screened these lineages for the presence and absence of proteins involved in key cellular processes. And so this included ribosomal proteins, topoisomerases, cell division and cytoskeletal proteins, as well as DNA repair, histones, and some replication factors. And this can kind of just be thought of as a barcode to distinguish the Culexarchaeota from other archaeal lineages, and we do see that they have a distinct pattern when compared with the closest relative Archaeota, as well as other archaeal lineages. And then in particular, they appear to have a very distinct set of cell division and cytoskeletal proteins within this phylogenetic space. And so that's a little bit on who the Culexarchaea are and how they relate to other archaeal lineages. And now we're wondering, what are these organisms actually doing in the environment? And so we reconstructed the metabolic potential from those 10 mags and found that they encoded a very diverse set of metabolisms. And this includes 4 hydrogenases, the potential for methylotrophy via the Wood-Jungdahl pathway, 5 potential electron bifurcating or conforcating complexes, carboxytrophy, beta oxidation, peptide fermentation, and sugar utilization, specifically fructose and galactose. And so these organisms can have quite the diet if they want. And to just focus in on one interesting story, so the Culexarchaeota encode some unique potentially heterodisulfide interacting nickel iron hydrogenases. And so on the left here is the phylogenetic tree of group 4 nickel iron hydrogenases. And down in the bottom, we see that the Culexarchaeota, the Archaeota encode some unique hydrogenases that were related to those previously only seen in the Methanosolates. And then we also found that another close relative to the Culex in the Archaeota also encode one of these unique membrane-bound nickel iron hydrogenases. And the takeaway from these complexes is that they're modular in terms that they can interact, potentially interact with other subunits such as succinate dehydrogenase or carbon monoxide dehydrogenase in the Culex. And these may have been important for substrate diversification and energy conservation strategies in both methanogenic and non-methanogenic lineages. And so just a few conclusions. So the Culex are a non-methanogenic archaeal lineage that's most closely related to the Archaeota. They do have a distinct pattern of proteins involved in key cellular processes. They encode the potential to metabolize a wide variety of substrates, including sugars, proteins, fatty acids, hydrogen, carbon monoxide, and methyl compounds. And they encode unique modular nickel iron hydrogenases. And so in future work, we'd like to incorporate fish for visualizing these cells, other omics, including RNA and protein, as well as stable isotope probing, and of course, targeted enrichment cultivation. And so with that, I'd like to thank all the funding sources and members of the lab. And yeah, thank you.
Thanks, Anthony. All right, moving right along. Our next speaker is Thiberio Rangel from MIT. Thiberio is a computational evolutionary biologist that studies evolutionary trends and the intricacies of phylogenetic tree space. So if you are ready, Thiberio, take it away.
Yeah, thank you, Greg. Thank you for the opportunity to present the work. And yes, we're going to speak about my work and coincidentally Greg's work about the impact of fast evolving sites in the reconstruction of deep phylogenies, right? How informative they are to phylogenies. Okay. I'm imagining everyone can see the slides, otherwise I would have heard someone screaming so far. So the main idea is that different sites that are evolving at different speeds, right, at different rates, they accumulate substitutions across with different frequencies as expected, right, as the name intends. And with these different frequencies, these different rates of evolution, they can be informative at different levels across different branch lengths. Sorry, let me just try to remove this window. Yeah, sorry. Yes. So our idea is that you have sites in an alignment that are evolving at different speeds. And once we characterize them, we bin them at different substitution rate categories, we can try to evaluate how informative those sites are to the reconstruction of the final tree. So our hypothesis is that fast evolving sites are the only ones or the most likely to be informative across short branch bipartitions. And sadly, those sites are the same ones that are frequently trimmed from alignments during slow fast analysis that tend to remove fast evolving sites because they are likely saturated, right? They have saturated substitutions. And the problem is that the remaining slow evolving sites likely did not experience substitutions during the short period of time represented by short branch bipartitions. So to test it, we used this small unknown study from Hooff, Laura Hooff, right, from 2016, very well known. And we got this, the alignments, the multiple sequence alignment. We classified all the sites in 12 substitution rate categories. And we used those sites from each of those substitution rate categories to reconstruct the original tree to see how close they were to reconstruct the final tree, right? Using sites only for each specific substitution rate category. We also redid this using a simulated alignment, a simulated tree, simulated phylogeny, and then we would have a known underlying phylogeny to which we can compare to. Because while the Hooff tree is a good approximation, we do not know the real underlying phylogeny, right? And with that, the result that we got were close to what we expected in some form, but very, very interesting in the sense that we can see here in the left, right, this is an MDS, multidimensional scaling, with the original tree from the whole alignment represented by this green X, and the cloud of trees the cloud of trees obtained from each substitution rate category specific phylogenies close to it. And you see that the trees from mid substitution rates, right, from the median rates of substitution are the closest to the whole alignment tree, while you see that the worst ones, or the ones that are less similar to the whole alignment, are from the slowest evolving sites, which are these green circles, and the fastest evolving sites. But the main problem is that the slowest evolving sites are still far away, and less consistent. The other thing that you see is that from our simulations, I don't know if you can see it because something's in the middle here of my screen. We see that for the simulations, we see that the slow evolving sites perform significantly worse than the fastest evolving sites in general, even when considering a true known known tree, and which was reconstructed using a true or the most correct as possible evolutionary model. When we look at what are the branches, the bipartitions that were most likely misreconstructed, what we see is that the slow evolving sites, the slowest evolving sites, were not able to reconstruct the shortest bipartitions. We see in the y-axis here is the branch length of the correctly reconstructed bipartitions from the Hooff tree, and what we see is that the slow evolving sites are not able to reconstruct those bipartition that have shorter branch lengths. In this dashed line, you can see the median branch length. So you see that there's the tree as a whole has fairly short branch lengths, and the slow evolving sites fail to reconstruct those. And you see as this evolutionary rates get faster, the sites are able, the site-specific phylogenies are able to reconstruct those short branched bipartitions. And you see the result is replicated by the simulations fairly well. Another thing that we saw is that the slow evolving sites, they tend to have a strong compositional bias. We see here that the two slowest evolutionary substitution rates, they have very biased composition and they group with themselves here and completely away from the other substitution rate categories. And with the whole tree alignment, using this alignment, just by changing the expected composition, right, from the default from LG to an empirical composition, adding just the F to the model, we obtain completely different phylogenies, right? Even getting to the point that we have a 3-domain of life tree just by changing the amino acid frequency. That is to show how impactful those subjective trimmings can be across different evolutionary models. Here we have just a bunch of examples, and you see that using different trimming strategies and different evolutionary models, we have completely different results. And just to close up, just want to show that as we increase, as we get more more species, as we increase the number of taxa used for our phylogenies, the branch lengths of our trees tend to decrease. So as in the future, the importance of the fast evolving sites are going to be even higher, even greater, because the branches will tend to get shorter and the time spent shorter as well. I just want to thank Greg and the funding agencies and the opportunity to be here. Thank you very much. Talk to you soon.
Great, thanks, Thiberio. And then we'll move on to our final of the short contributed talks from Jolien van Hooff from ESC, CNRS, University of Paris-Saclay, AgroParisTech. Jolien is a postdoctoral researcher at the CNRS in Paris in the group of Dr. Laura Aimé. Her work broadly comprises genome evolution across the eukaryotic tree of life. So we'll get to hear about some eukaryotes too today.
Yeah, thanks for the introduction, Craig. Yes, I guess I'm a bit of an outlier here because indeed I'll be talking today about eukaryotes, more specifically about lateral gene transfer events, and more specifically even about these events in Rhizaria. And Rhizaria is actually what you can see right here in the background of my presentation. I'll tell more about these species later in my presentation. So we are interested in studying lateral gene transfer in eukaryotes basically because it's still quite an enigmatic phenomenon. And that's in part due to the fact that for many eukaryotic clades, there's still only few sequence data available. And also because in contrast to in prokaryotes, there's no well-defined mechanisms for lateral gene transfer in eukaryotes. So for these reasons, for a long time, lateral gene transfer was considered virtually absent in eukaryotes. But over the last decade or so, many convincing cases on LGT into eukaryotic genomes have been published. So all the more reasons to study this phenomenon more broadly and answering questions like how frequent is lateral gene transfer in eukaryotes? From which species to which species are they being transferred? Which functions are transferred? Where in the genomes do the LGTs localize? What could be mechanisms of lateral gene transfer in eukaryotes? And might they be drivers of evolutionary transitions? Now, as I said, we are currently studying this specifically in Rhizaria, and that's mainly because these species are highly abundant in the oceans. They're mostly free-living unicellular heterotrophs and thereby a good model for many eukaryotes broadly. There are some photosynthetic lineages and also some pathogens. Altogether, that suggests that there has been various evolutionary transitions in the Rhizarian tree of life, and potentially also LGTs were involved in those. Another reason for studying Rhizaria is that up until now, they are very poorly studied. And this is, for example, illustrated by the fact that up until today, only 3 published genomes are available. So by studying lateral gene transfers and genome evolution in these species more broadly, we hope to be able to get to know them a bit better. So as you can see right here, Rhizaria are actually a pretty old clade in the eukaryotic tree of life, and they group with the Terminopals, which, for example, encompasses the diatoms, and with the alveolates, which, for example, includes dinoflagellates. So using a phylogenomics approach, we studied LGT events in 30 Rhizarian species, as indicated here on the x-axis. And we established that for the genes for which we were able to determine this, about 32% on average was derived from a lateral gene transfer. And for most of the species, as you might see right here, the percentage is somewhere between 25 and 45%. Although obviously there are some clear outliers, such as this Glypha rotunda species. If we go a bit deeper into these LGTs, we can see that not all of them do actually comprise a given LGT in that particular lineage. Some of them are actually older than the lineage itself, so they are shared among multiple Rhizarians. And so insofar as contamination is a risk of erroneously detecting LGTs, that at least does not apply to these shared LGTs. So if we project all of the LGTs that we detected onto the Rhizarian species tree, we can see that, for example, right here, there's a deep branch in here which gained many genes, nearly 400, via LGT. And I think that's a particularly interesting one because actually this is a highly biodiverse group, these sarcozoans. So it's kind of difficult to speculate what their sort of the biological significance of these LGTs might have been. Studying the putative donors of the LGTs, we see that most of them were likely derived from bacteria. However, also eukaryotes likely contributed various genes to these Rhizarian genomes. However, it's important also to note that these donor distributions differ quite a bit for specific lineages. For example, if we only take the lateral gene transfer events into Leptophrys vorax, one of the 30 species in our database, we can see that the plants contributed much more genes relative to the overall distribution. So such specific donor patterns for specific species might also indicate given potential species-species interactions. We were also interested in finding out what makes laterally transferred genes different compared to vertically inherited ones. Now, these genes mostly encode proteins that are somewhat shorter. They have fewer cortical regions, fewer intrinsic disorder, and fewer PFAM domains. Now, obviously, these might all be correlated to one another. That would make perfect sense. But one can also imagine that these features also make a gene somewhat more transmissible. If we look at the overrepresentations of predicted protein localizations, we see that the LGTs often localize to mitochondria, to the extracellular environment, and to the peroxisome. And well, as far as we know, for example, the bias towards the mitochondrion hasn't really been observed so far. Now, if we look at putative functions for the LGT, we see that they are relatively often involved in cell motility, in defense mechanisms, and in various metabolism-related functions. And in here again, particularly the first one, cell motility, and so far as I know, that hasn't been reported yet. So what are our next plans? Well, we aim to study the genomic localizations of these LGTs, as I said, for the species for which we have genomes. And we aim to shed light on how LGTs might be somehow correlated to the ecologies of the species. And with that, I'd like to finish my presentation, and I'm happy to answer any questions later in the breakout room.
Thank you. Yeah, and just with that, one additional reminder that you can ask questions for all of these, from all these contributed speakers during the breakout session. And so now we have our final speaker of the day, Tom Williams from the University of Bristol, UK. Tom is a Royal Society University Research Fellow and Associate Professor in Molecular Evolution in the School of Biological Sciences at University of Bristol. His research focuses on deep phylogeny using phylogenetics and comparative genomics to study early life and early cellular evolution. Okay, Tom.
Thanks, Greg.
Your little Koopa kid sticker in the corner. That's great.
Yeah, yeah. Can you hear me okay? I guess you can. Okay, let me know if you can't hear me. Anyway, yeah, thanks very much for the invitation. I'm very honored. I'm a little bit tired speaking to you tonight, but it's great. I'm going to talk about some work on bacterial phylogeny that I've been doing collaboratively with some other people. And I'll talk about some recent work on, I guess, trying to infer the roots of the bacterial tree of life and the nature of the last common ancestor bacteria. And I'll also tell you about some new follow-on work that we've been doing. And before I get into it, I just want to sort of acknowledge that this is a very collaborative program. So it's been a collaboration between my lab at Bristol, but also the lab of Anja Spang at NIOS in the Netherlands, Phil Hugenholtz in Brisbane, and Gergely Solosi in Budapest, working on methods. And a lot of what I'm going to talk about can be found in a recent paper, Coleman et al., that we published. So I don't think I need to convince this audience that bacterial evolution is interesting, but if you're ever talking to a paleontologist and you want to sort of impress on them why that's the case, then these could be the talking points. So, you know, bacteria are among the most diverse cellular organisms on the planet today. And as far as we can tell, that's been true if you sort of go back into the deep past. This sort of iconic tree from the landmark study of Hugg et al. Shows you that, you know, most genetic diversity seems to be prokaryotic, and maybe the majority of that is bacterial. So if you want to have an account of life's evolution or the tree of life, you can't sort of put microbes aside, right? I mean, perhaps everyone's convinced of that. From the evolutionary biology perspective, of course, there's been so many new groups discovered recently by environmental genomics. And there's some pretty open and very basic questions about bacterial evolution that I'm not sure that we've really answered completely. And what we've been trying to do is work on some of those, and in particular, two of them. So is there a tree of bacteria? So some people have suggested that there's so much gene transfer as you go deeper into bacterial evolution that it's almost misleading to think of, you know, a phylogeny of bacteria. If there is a tree, where's the root? And what was the last bacterial common ancestor actually like? What sort of traits did it have? So I'll tell you about some of our attempts to address those questions. And I'm going to talk about three major groups, or major candidate groups, if you like, of bacterial diversity in the talk. The first is the Pterobacteria. So that's a group that contains the Firmicutes, the Actinobacteria, Cyanobacteria, and their relatives. And the concept of that name originally comes from the idea that all of these lineages ancestrally have some adaptations to perhaps life on land or life in a less aqueous environment. You don't necessarily have to buy into that concept to acknowledge that there's, you know, a lot of evidence that at least some of these lineages are related evolutionarily. And then the second is the Gracilicutes. So those are the traditional double membrane gram-negative bacteria, so Proteobacteria, Bacteroidetes, the PVC group, and their relatives. And then finally, the candidate phyloradiation, or CPR, or sometimes called Patetsky bacteria as well. So that's a very recently discovered group of bacteria that seem to be reduced, or at least they have small cells, small genomes, and they may be ectoparasites or symbionts of other bacteria. So the interrelationships of these three lineages, if you like, will be sort of the subject of the talk. So then taking a step back, you know, this is pretty basic stuff in some sense, but, you know, what is the difficulty with inferring a tree of bacteria? And we already heard some of the challenges of inferring deep phylogeny in this symposium, but, you know, the first thing is that ancient divergences of any kind are going to be difficult to reconstruct because we're talking about things that happened a long time ago. And recent changes in evolution may have overwritten the evidence and sequence data for those deep relationships. For bacteria, we've also got the issue that there's been a lot of gene transfer. So what genes should we use to reconstruct the tree? And does that gene transfer have the potential to blur or mislead our inference of deep relationships? And then there's a third issue, which is that normally when we root phylogenetic trees, we use an outgroup. But for deep relationships, like between cellular domains, that creates difficulties. So if you want to root the bacterial tree with an outgroup, your only choice is to use the Archaea. But if there's sort of 2,000 to 4,000 genes on an average bacterial genome, then there's really 30 to 40, depending how you count it, genes that are shared between bacteria and Archaea that may have evolved vertically since their common ancestor. So you're immediately boiling down to a very small proportion of the available data to resolve those relationships. And as well as that, if you look at the structure of the tree here, I think you can see my mouse, then the branch that connects bacteria to Archaea is probably the longest one in that tree. And if the question you're asking relates to the deep splits here, and again, we already heard a bit about the difficulty of recovering some of those rather short branches, the long branch can cause problems with inferring the short branch relationships, such as long branch attraction artifacts. So if we had a way of rooting the tree without an outgroup, that might bring new insight to the problem. It doesn't mean we can't use outgroup rooting to look at the deep relationships between Archaea and bacteria. We can, but other methods might be interesting and give complementary insights. So there's a few different approaches that people have taken here, and I'm going to show you actually evidence from all three, although for two of them very brief. So one approach would be to use a molecular clock or something like a molecular clock assumption. So the molecular clock is the idea that genetic divergence accumulates roughly proportional to time. So if you have two lineages or two organisms that diverged a long time ago, then they're going to have accumulated more genetic change than two lineages that diverged recently. You could use a full molecular clock model, or there's methods like MAD, MinVar, which use branch length information to try to root a tree. So if you root a tree somewhere in the middle, that could give a good estimate of the tree. There's also non-reversible substitution models. So the normal phylogenetic models that we use don't intrinsically root trees because we normally don't try to capture evidence about trends or the direction of evolution. But for example, Greg Fournier's work has shown that there may be trends in amino acid compositions through time. So in the past, perhaps there was fewer amino acid positions occupied by tryptophan than there are today. So if we had a model that could try to capture changing trends in amino acid composition or other signals that vary with time, we might be able to root a tree without using an outgroup. And then we can also use gene tree, species tree reconciliation. And that's a direction that I've been interested in and working on for a while. So I'm going to talk about that in a little bit more detail. So this is the basic setup for probabilistic gene tree, species tree reconciliation. I'll explain what it is, and then I'll tell you why we can use it to root the tree of bacteria. So the data are gene family alignments, like in normal phylogenetic analysis, right? And normally you would have a substitution model that describes how the sequences of your alignment have evolved on some kind of tree structure, experiencing substitutions along the way. We can infer the tree and the substitution history using like maximum likelihood or a Bayesian method. What we're trying to do with gene tree, species tree reconciliation is add an extra layer to this process, where as well as having a substitution model describing how the sequences evolve on the tree, we have a gene tree, species tree reconciliation model that describes how the gene trees evolve on the species tree, occasionally experiencing events like, for example, transfers between different branches. And again, we can use maximum likelihood or Bayesian methods to try to find the rooted species tree that best explains the observed gene family alignments. And the sort of biological intuition here, or one way of talking about this would be to say that, in some sense, it's an extension of the classic studies on anciently duplicated genes for rooting the tree of life. So, Iwabe and Gogarten in 1989 and their collaborators showed that if you look at anciently duplicated genes, you can root the tree of life by using one duplicate as a sort of a mirror tree, or one duplicate to root the other. So that's the evidence that the root of the tree of life is between bacteria and other cells. So can we extend this to the whole genome, but also use transfers and the gene loss events to try to infer where the root is? So in our recent work, we used one of these probabilistic gene tree, species tree methods to try to find the root of the tree of life. Okay, sorry. Before I show you the results, I'll just talk very briefly about method validation. I mean, this is a new approach. Does it work? So in simulations, it works pretty well. For example, if you simulate gene alignments on a rooted species tree, then in our study for 95% of replicates, or for 19 of 20 replicates, the maximum likelihood root was the true root. And in the case where it wasn't, the inference from the method was one branch away from where the true root was. If you ask, can these methods recover the simulated number of duplication events, transfer events, or loss events? The answer is they do fairly well at recovering the true number of events, possibly with the exception of losses, where when you have a lot of losses, then the current implementation of the methods in the AL package, which is the one that we're using, we slightly underestimate the number of simulated losses. So methods seem to work okay. What happens when we apply them to genome data? So here we use 265 genomes sampled from the Genome Taxonomy database. And we place the root between the Gracilicutes, so the classic double membrane bacteria, and the Terrabacteria, so that's Firmicutes, Cyanobacteria, Actinobacteria, and their relatives. And in contrast to some outgroup rooted studies, in these analyses, the CPR group actually branched within the Terrabacteria, a sister to the clade of Chloroflexota plus Dormibacteriota. We were unable to replace Fusobacteriota. We weren't sure whether this group goes with Gracilicutes or with Terrabacteria, but nonetheless, we were fairly sure that the root's somewhere between Gracilicutes and Terrabacteria. So I mentioned at the start, there's this question of how tree-like is bacterial evolution, or is there a tree of bacteria, at least at the deepest splits? And maybe you think I put the cart before the horse by showing you this rooted phylogeny inferred using gene tree, species tree reconciliation, but actually testing whether there's much tree-like signal in the data. But fitting a model of this kind actually gives you some empirical grasp on that question as well. Because if we take that rooted species tree that we inferred with the gene tree, species tree reconciliation model, we can ask how many of the branches of the gene trees that we use, so 11,272 gene trees, how many of those map onto the species tree? In particular, if we consider like a one branch of the species tree here from its sort of ancestral node to the descendant node, how many of the events that we see on that branch are vertical descent from the ancestor to the descendant compared to the origination of a new gene or the acquisition of a gene by transfer from another lineage? And over 2 replicate data sets, the average was 66% of the gene transmissions were vertical. So that suggests that a rooted species tree is quite a good summary of a significant vertical component of bacterial evolution. It's not as simple though as saying, oh, well, this means that there's, this debate about is it a tree or is it a network is resolved one way or the other, because it sort of depends on your perspective or the way that you want to view things. So 66% of gene transmissions are vertical. That means if you take a slice through the time tree of bacteria, at any one time, there's a lot of vertical evolution. But if you actually look at an individual gene family, what you see is that it's almost certain to have experienced gene transfers throughout its history, right? So in this analysis, we take gene families in modern genomes, and we try to trace them back along that rooted species tree, and we ask how many of them get to the root. And if they don't get to the root, where was it in evolutionary history that they experienced like a non-vertical event, a transfer event? And actually the great majority of gene families in this analysis experience transfer before they get to the, here we're going back in time, right? So before they get to the crown of the phylum they're in. So most gene families on modern bacterial genomes have been transferred onto that genome lineage, if you like, since the crown of the phylum. And then there's a smaller proportion of genes which trace further back vertically, and that's the sort of traditional genetic core, plus a bit more maybe that, that people have used in concatenation studies and other studies in the past. So that's 2 different ways of looking at the verticality of bacterial evolution. Okay. I also wanted to update you on some new analyses. So we use 265 genomes here. We want to scale that up now that we've got more and more environmental genomics data. It turns out to be quite difficult for methods like ALE, so the probabilistic gene tree, species tree methods, but we can use other routing methods to ask, actually, when we look at more data, do we see the same route? And we seem to, at least in our analyses. So this is analysis of 1,007 genomes sampled from the Genome Taxonomy database. And the tree I've shown you here is routed using the MAD method. So that's a method that's based on the idea of a molecular clock. It uses branch lengths to try to route the tree. And we root in the same place, right, between Gracilicutes and Terrabacteria. We also have been using a non-reversible model that's implemented in IQ tree, so non-rev plus G, which tries to capture trends in amino acid composition. It also routes on the same branch between Gracilicutes and Terrabacteria on this data set. So there seems to be some consistent signal here as we try to scale up. But these methods are less certain about where the route is than the gene tree species tree methods, in part perhaps because they're using less data. So they're using concatenate of 65 genes in this case. So here the colors of the branches reflect how suboptimal a route on that branch would be according to MAD's route criteria. And what you can see is really, according to the MAD method, a number of different branches deep in the tree would fulfill the routing the optimization criteria almost as well. So the optimal position is the same as what we get with the gene tree species tree analysis, but moving the route around, at least on those deep branches, wouldn't affect the result too much. So it's preliminary analyses. A number of outgroup routing studies have been published that are relevant for understanding the position both of the CPR and of where the root of bacteria might be. And I think maybe the conclusion from the outgroup routing studies that have been published recently is that there is a consistent signal for the CPR grouping with chloroflexy. But as for the position of the root, there's one outgroup routing analysis, Rayman et al. 2015, that found a route between those two groups. But I think there's a lot more opportunity for research and lots of interesting stuff to be done there before we find out whether this is a result that's supported from many different kinds of analyses. And I wanted to finish really by talking about what gene tree species tree reconciliation can suggest about the nature of the last bacterial common ancestor. So here, and this is also part of that recently published study, we created gene families based on COG clusters. So that's a functional ontology of gene families. And then we mapped these COG families to the root of the tree using the same approach as we used to root the tree. And we inferred for each family a root presence probability. And then based on families with high probabilities, we reconstructed a sort of a cartoon of what the last bacterial common ancestor might have been like. Based on this analysis, it would have had a genome size of between 1.6 and 2.7 megabases, not fairly modest for a free living bacterium maybe. In terms of its metabolism, it's actually difficult to say because we saw support for the reverse TCA or the TCA cycle, as well as pathways like the Wood-Lungdal pathway, which we also heard about this evening. But based on the presence of an RNF complex and some components of the Wood-Lungdal pathway, we think it might have been an acetogen, so capable of acetogenic growth. It was probably rod-shaped based on having some of the key genes for that phenotype. It had a flagellum, it was capable of chemotaxis, it had a CRISPR-Cas system, and it seemed to have been double membraned based on a number of genes involved in, for example, lipopolysaccharide biosynthesis. And of those different traits, many of those agree with various studies that have been published before. The idea of a double membrane or diderm last bacterial common ancestor was sort of a little bit surprising or interesting to me, although it's consistent with some previous studies. And one thing that's interesting, I've taken this figure from a recent by Witwineski et al. Is that actually if this route between Gracilicutes and Terrabacteria is correct, then if you look at the distribution of a single or double membrane across modern bacteria, then it's sort of not so surprising that the last common ancestor would have been a diderm, because most groups on either side of the root, certainly all the Gracilicutes, but also the majority of the Terrabacterial groups appear to be diderms, with monodermy or gram-positive phenotype evolving several times independently within the Terrabacteria. But that's something, again, that's going to be really interesting to pursue as we get more data, and also considering like different methods we could use to try to reconstruct ancestral traits. So the biological conclusions of my talk, so it seems like based on these outgroup free rooting methods, that the rudobacteria between the Gracilloceutes and the Terrabacteria, so two major clades, that the CPR bacteria or the Patetsky bacteria seem to be derived from within the Terrabacteria. That the ancestor was a rod-shaped flagellated cell with a double membrane, and also that we can try to model both the vertical and horizontal components of bacterial evolution jointly in a kind of a gene tree, species tree framework. And I sort of finish with a little bit of outlook. I mean, part of the motivation here for doing gene tree, species tree reconciliation, and I know some of the people here are sort of also really interested in these approaches, is that we can combine phylogenetics and comparative genomics, or at least sort of we can have a model which tries to use as much of the data, genome data, as possible to reconstruct the evolutionary history, but at the same time sort of map the origin of genes and traits. So there's a lot of promise in that framework, I think, but the models that we have now have limitations, and we're working on making them better, and I think other people are doing that as well. So methods for inferring gene families from genome data automatically are not consummate. Here we use two different approaches, the COG approach and the MCL approach, but presumably there's something better. Reconciliation models are fairly simple. There's parsimony ones, there's probabilistic ones. The probabilistic ones, for example, ALE has one duplication transfer and loss rate for a family or for a set of families, whereas presumably those rates should vary across the branches of the tree, for example. Scaling to large data sets is difficult, and that's frustrating because we're getting so much more data being published from environmental genomics that presumably contains the information we need to answer these questions about early evolution, if we can harness it. And even, we're building something here on top of substitution models, and we know that substitution models could use some work as well. So there's a lot of scope for improvement at every step of the pipeline, but I think there's also real opportunities to make these things better and learn more as we go. So thanks very much for listening to my talk. I'm very happy to chat in the breakout session. Yeah, thank you.
Thanks, Tom. And you finished exactly with 5 minutes for questions.
Oh, good.
So I don't see any questions posted in the chat right now. People may have forgotten that since we weren't taking questions for the previous short talks. But in the next 5 minutes before we go to the breakout rooms, please post any questions you have for Tom here. And while people are thinking about that, I have a question.
Sure.
So our group has also thought a lot about what we can do using HGT and resolving tree of life and disentangling these questions. And the last point you made, the simplicity of reconciliation models. I'm curious as to your thoughts about the general philosophy behind reconciliation, which appears to be the notion that there is some kind of argument of parsimony behind the true history of a gene, so that minimizing the number of events needed to fit a gene tree to a species tree is likely to converge on that true history. And you briefly mentioned, that these models are simple because the rate of DTLs or the potential frequency can change across branches. But can you see a way that also may vary across genes? And how could we better model that information, given that we have some knowledge that different groups of genes may have a different prior belief in being transferred versus lost versus duplicated?
Yeah, that's a really good question. I think there's a couple of different points that I would like to make. So one is, you mentioned the philosophy of reconciliation and the idea of trying to minimize the cost between reconciling the gene tree to the species tree. I think, with the probabilistic methods, while they're simple now, I'm not sure that's necessarily the underpinning philosophy. you might have a model view where you have like a generative model. So you have like, there really is a species tree or a tree of cells or some kind of multi-level evolution framework of some kind. And you really are evolving genes like on a species tree, but they occasionally undergo different sorts of events. And it's not necessarily the case that the true reconciliation will be the one that minimizes the events. There can be heterogeneity in the rates and so on. So like in a philosophical sense, I think, you know, you can imagine a very complicated model underpinning all of this. And what we're trying to do is like estimate the parameters of that model or do inference with that model. The more practical question, how will we actually make the models better? Well, you suggested one approach. So, you know, we know that we expect that genes involved in translation are transferred less. So we could have like rates that differ across the categories of genes. And for inferring the root probabilities here, we tried to do that, or we did do that. So we had for each category, we estimated a separate, independently estimated root origination parameter. We could do something like that for all of the parameters of the model. Ideally, we would have something like simply a gamma distribution of rates over branches on the tree, for example. That's difficult though, because we have like at least 3 parameters that we want to have a gamma distribution on. So if we have 4 categories, it gets computationally expensive. But in principle, that's the sort of thing we would want to do. Maybe there's a way of approaching that more cheaply by doing something that clusters the gene families together into sets, which are supposed to evolve in some kind of homogeneous way. And then we estimate the parameters separately for those different sets. That's something that we're pursuing at the moment. But whether it's going to be possible to do inference under those models in the way that we conceive them now, I'm not totally sure, but that's the kind of direction I would think of going.
Thanks, that's really interesting. I'm glad to hear that work's being done on it. So briefly, we have 2 questions from the audience before we go to breakout. The first from Anonymous is, would you say that single membrane bacterium are closer to eukaryotes than double membrane ones?
That's a good question. I've always been intrigued by some of those trees placing the root of the tree of life within bacteria and having like Archaea evolve from within the single membrane bacteria. And that's you know, I've always found those papers very interesting, but in terms of finding evidence from them, from the sorts of models that we have, I don't think there's much evidence or any. I don't want to say any because I may be forgetting something, but I think it may be that as we make these models better, we suddenly start finding very surprising things. But at the moment, the monophyly of bacteria seems reasonably well supported from analyses. Yeah, maybe it's a bit of an empirical answer to the question, but that's my view.
And our second question from Jean Fagy is there any other method that can be used or developed to detect HGT besides gene tree to species tree reconciliation? Can species trees still be reliably inferred based on concatenated genomic data when HGT is prevalent?
So there's 2 questions there. Is there a way of detecting gene transfer apart from gene tree species tree reconciliation? Yes, I think so. I mean, my impression is that most of the time when people, let's say you're writing a paper about an interesting HGT, most of the time you've detected that because either you formally had a species tree and the gene tree of the HGT conflicts with it, or it's not formal, but there's some kind of underlying idea of a reference tree and this gene conflicts with it, therefore it's an HGT. However, you can look at the composition of sequences. For example, if you have a sequence on a genome with a very weird composition, then that might suggest it's an HGT. If you're looking at eukaryotes, then genes which have unusual structural features, as Jolien was mentioning, that's a way of detecting HGTs. It's possible to infer networks. There are blast-based similarity networks. There's also a recent paper from Alexis Stamatakis where they're suggesting a method for estimating a network directly from gene data. So there are methods for estimating this non-vertical evolution without assuming a species tree. The second point, there's a sort of hinting at a circularity. So, you know, we concatenate the genes, we get the species tree, then we reconcile everything. But actually, when we reconcile the genes that made up the concatenate, do they actually evolve vertically? This is a really good question. I think there's a tension there. I suppose there are genes that are very rarely transferred, and those are the ones that we're using to make the concatenates. And I suppose that we hope that very occasional transfers in the concatenate are not going to seriously affect the topology that we get. The alternative, of course, is to do a tree search, or one alternative, I should say, would be to do a tree search as part of the reconciliation process. So here we did concatenation, and then we find the root with the reconciliation. But for example, species ranks is a method. It had a small role in its development, but it's basically developed by Stamatakis and his group, where you infer the species tree and the gene trees at the same time. And so you don't bias the species tree towards any particular genes. So there are ways of not assuming a species tree. But I would suggest that there may be genes that you can concatenate and get a pretty good estimate of the gene tree, of the species tree rather.
Great. Okay, well, we are out of time for questions here. I'm going to hand things over to Emily, and she will be opening breakout rooms.
Okay, great. Thanks so much. So I want to thank all of the speakers. That was just fabulous. I am super excited thinking about the first NeLLi symposium that we had back in 2015. It's just amazing the amount of progress, especially on the tree of life just within the past 6 years. So thanks to everyone for joining us. Special thanks to Greg for moderating.